What is gluconeogenesis? Gluconeogenesis is the metabolic pathway that generates glucose from non-carbohydrate carbon substrates. These non-carbohydrate substrates include lactate, amino acids, and glycerol, which can be converted into glucose when needed. Gluconeogenesis is an anabolic process, which means it builds up new molecules rather than breaking them down. Let's compare gluconeogenesis with glycolysis. While glycolysis breaks down glucose into pyruvate, gluconeogenesis does the opposite. It creates new glucose molecules from substrates like pyruvate. Gluconeogenesis is crucial for maintaining blood glucose levels, especially during fasting periods or when carbohydrate intake is limited. Without this process, the body couldn't maintain normal blood glucose levels during extended periods without food. In summary, gluconeogenesis is the metabolic pathway that creates new glucose from non-carbohydrate sources, serving as a critical mechanism for maintaining blood glucose homeostasis. Why is gluconeogenesis important? The brain and red blood cells primarily depend on glucose as their main energy source. Unlike other tissues, the brain cannot efficiently use alternative fuels like fatty acids, making glucose essential for normal brain function. Maintaining blood glucose levels within a normal range is essential for brain function and survival. When blood glucose drops too low, a condition called hypoglycemia, it can lead to confusion, loss of consciousness, and even death if severe and prolonged. Gluconeogenesis becomes increasingly important during fasting or starvation. In the first few hours without eating, the body relies on glycogen stores. But as fasting continues beyond 8 to 12 hours, glycogen stores deplete and gluconeogenesis becomes the primary source of blood glucose, particularly for the brain. Gluconeogenesis is a metabolic adaptation that allows the body to maintain blood glucose levels during various nutritional states, including low-carbohydrate diets, exercise, and fasting. From an evolutionary perspective, gluconeogenesis is a critical adaptation that allowed our ancestors to survive periods of food scarcity and varying food availability. This metabolic pathway ensured that even when carbohydrates were scarce, the brain could still receive the glucose it needs to function properly. The liver is the primary site of gluconeogenesis, responsible for approximately 90% of glucose production in the body. Liver cells, called hepatocytes, contain all the necessary enzymes for the complete gluconeogenesis pathway. The liver's anatomical position receiving blood directly from the digestive tract makes it ideally situated for gluconeogenesis. The liver plays this central role because it can efficiently capture and process gluconeogenic precursors from the bloodstream, including lactate, amino acids, and glycerol. While the liver is the primary site of gluconeogenesis, the kidneys serve as the secondary location for this crucial process. Under normal conditions, the kidneys contribute approximately 10% of total glucose production through gluconeogenesis. However, during prolonged fasting, the kidneys' contribution becomes significantly more important, increasing to 20 to 25% of total glucose production. The renal cortex, the outer portion of the kidney, specifically contains all the necessary enzymes required for gluconeogenesis. Other tissues, such as the intestinal mucosa, can perform limited gluconeogenesis. However, they are considered minor contributors to overall glucose production compared to the liver and kidneys. Understanding the secondary locations of gluconeogenesis, particularly the kidneys, is important for comprehending how the body maintains blood glucose levels during different physiological states. Glycolysis and gluconeogenesis are opposite but related metabolic pathways. Glycolysis is a catabolic pathway that breaks down glucose into pyruvate, releasing energy. Gluconeogenesis is an anabolic pathway that builds glucose from pyruvate, requiring energy input. Looking at both pathways in detail, we can see they share many intermediate steps. In glycolysis, the pathway flows downward from glucose to pyruvate, while gluconeogenesis runs in the opposite direction. Gluconeogenesis cannot simply reverse glycolysis because 3 steps in glycolysis are irreversible. These irreversible steps must be bypassed using different enzymes in gluconeogenesis. The two pathways also differ in their energy profiles. Glycolysis produces energy, while gluconeogenesis requires significant energy input. To summarize, while glycolysis and gluconeogenesis involve many of the same intermediates, they differ in direction, energy requirements, and the enzymes used at key irreversible steps. Lactate is an important substrate for gluconeogenesis, particularly during and after exercise. The Cori cycle is a metabolic pathway that connects skeletal muscle and the liver, allowing lactate produced during anaerobic exercise to be recycled back into glucose. During intense exercise, muscles shift to anaerobic glycolysis, converting glucose to pyruvate and then to lactate. This lactate is then released into the bloodstream and transported to the liver. In the liver, lactate is converted back to pyruvate by the enzyme lactate dehydrogenase, or LDH. Pyruvate then enters the gluconeogenesis pathway, ultimately producing glucose, which requires energy in the form of ATP. The newly synthesized glucose is released into the bloodstream and returned to the muscles, completing the cycle. Let's examine the conversion of lactate to pyruvate more closely. Lactate dehydrogenase catalyzes the oxidation of lactate to pyruvate using NAD+ as a cofactor to accept electrons. Once converted to pyruvate, the molecule enters the gluconeogenesis pathway in the liver. The Cori cycle offers several important benefits. This recycling process is particularly important during intense exercise, when muscles produce large amounts of lactate, and during recovery, when the body needs to restore glucose supplies. During periods of fasting, triglycerides are broken down in a process called lipolysis. This breakdown releases glycerol and 3 fatty acids. While the fatty acids are primarily used for energy, glycerol can serve as a gluconeogenic substrate. The glycerol released from triglyceride breakdown enters gluconeogenesis through a multi-step process. First, glycerol is phosphorylated by the enzyme glycerol kinase to form glycerol 3-phosphate. This reaction requires ATP. Next, glycerol 3-phosphate is oxidized by glycerol 3-phosphate dehydrogenase to form dihydroxyacetone phosphate, or DHAP. DHAP is a direct intermediate in the gluconeogenesis pathway, connecting to the later steps that lead to glucose production. During fasting states, hormones like glucagon and catecholamines stimulate lipolysis in adipose tissue. This results in increased glycerol release, which provides an important substrate for gluconeogenesis when carbohydrates are limited. In summary, glycerol from lipid metabolism forms an important link between fat breakdown and glucose production, helping to maintain blood glucose levels during periods of fasting. Glucogenic amino acids are an important substrate for gluconeogenesis. Glucogenic amino acids can be metabolized to create glucose through the process of gluconeogenesis. These amino acids enter the gluconeogenic pathway at different points after undergoing deamination. Let's examine the key intermediates where amino acids can enter the gluconeogenic pathway. Amino acids like alanine, serine, glycine, threonine, and cysteine are converted to pyruvate. Aspartate and asparagine are converted directly to oxaloacetate. Glutamate, glutamine, arginine, proline, and histidine form alpha-ketoglutarate. Methionine, isoleucine, and valine are converted to succinyl-CoA. Phenylalanine and tyrosine enter as fumarate. Before amino acids can enter the gluconeogenic pathway, they must undergo deamination, the removal of their amino group. In deamination, the amino group is removed from the amino acid, producing a keto acid. The amino group enters the urea cycle while the carbon skeleton continues into gluconeogenesis. During prolonged fasting, amino acids from protein breakdown become increasingly important for gluconeogenesis. In the fed state, amino acids contribute minimally to glucose production. As fasting progresses, glycogen stores deplete, and amino acids provide more carbon skeletons for gluconeogenesis. During prolonged fasting, protein breakdown accelerates, and amino acids become the primary gluconeogenic substrate. Branched-chain amino acids, leucine, isoleucine, and valine, have a unique metabolic fate. Unlike other amino acids, branched-chain amino acids are primarily metabolized in muscle tissue, not the liver. After initial processing in muscle, their carbon skeletons are released into the bloodstream. These carbon skeletons then travel to the liver, where they can enter the gluconeogenic pathway. The first unique step of gluconeogenesis is the conversion of pyruvate to oxaloacetate. This reaction takes place in the mitochondria of the cell. The reaction converts pyruvate, a 3-carbon molecule, to oxaloacetate, a 4-carbon molecule. This reaction is catalyzed by the enzyme pyruvate carboxylase. The reaction requires ATP as an energy source and carbon dioxide as a substrate. Pyruvate carboxylase requires biotin as a cofactor, which serves as a carrier for the carbon dioxide molecule. The reaction proceeds in 2 steps. First, ATP activates the bind cofactor on the enzyme. Second, the activated carbon dioxide is transferred from biotin to pyruvate, forming oxaloacetate. Acetyl-CoA, derived from fatty acid oxidation, acts as an allosteric activator of pyruvate carboxylase. This creates an important metabolic link between fatty acid oxidation and gluconeogenesis. When fats are being broken down, producing acetyl-CoA, the body increases glucose production from pyruvate. The overall reaction converts pyruvate to oxaloacetate using 1 ATP molecule and carbon dioxide. This is an energy-requiring step that commits the cell to gluconeogenesis. After forming oxaloacetate, the gluconeogenesis pathway continues with the conversion to phosphoenolpyruvate in the next step. The second step of gluconeogenesis involves transporting oxaloacetate from the mitochondria to the cytosol. Oxaloacetate is produced in the mitochondria during the first step of gluconeogenesis. Since oxaloacetate cannot easily cross the mitochondrial membrane, it can be transported by 2 main pathways. It can either be transported directly in some cases, or more commonly, it is first converted to malate, which can freely cross the membrane. Once in the cytosol, oxaloacetate is ready for conversion to phosphoenolpyruvate. The conversion is catalyzed by the enzyme phosphoenolpyruvate carboxykinase, or PEPK. This enzyme requires GTP as an energy source. PEPC catalyzes the decarboxylation of oxaloacetate to form phosphoenolpyruvate, releasing carbon dioxide. During this reaction, GTP is converted to GDP, transferring a phosphate group to form phosphoenolpyruvate. This reaction is critical because it bypasses the irreversible pyruvate kinase step of glycolysis. PEPK is a major regulatory enzyme in gluconeogenesis. Its expression is controlled at the transcriptional level by several hormones. Glucagon, which is released during fasting, increases PEK expression. Cortisol, a stress hormone, also upregulates PEK expression, promoting gluconeogenesis. In contrast, insulin, which is abundant after meals, decreases PEK expression, inhibiting gluconeogenesis when glucose is plentiful. To summarize this critical step, oxaloacetate is transported from the mitochondria to the cytosol, where PEP converts it to phosphoenolpyruvate using GTP. This bypasses the irreversible pyruvate kinase step of glycolysis, and PEP's expression is tightly regulated by hormones to control the rate of gluconeogenesis. Steps 3 through 8 of gluconeogenesis use the same enzymes as glycolysis, but run in reverse direction. This pathway converts phosphoenolpyruvate to fructose 1,6-bisphosphate through a series of reversible reactions. Unlike the previous steps we discussed, these reactions are bidirectional, meaning they can flow in either direction. Step 3 involves the conversion of phosphoenolpyruvate to 2-phosphoglycerate, catalyzed by the enzyme enolase. In step 4, phosphoglycerate mutase converts 2-phosphoglycerate to 3-phosphoglycerate. Step 5 requires ATP and uses phosphoglycerate kinase to convert 3-phosphoglycerate to 1,3-bisphosphoglycerate. In step 6, glyceraldehyde 3-phosphate dehydrogenase utilizes NADH to convert 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. Steps 7 and 8 involve aldolase and triose phosphate isomerase, which convert glyceraldehyde 3-phosphate to fructose 1,6-bisphosphate. These reversible steps can proceed in either direction. The actual direction depends on several key factors. First, substrate concentration gradients push reactions toward product formation. Second, The cell's energy status, reflected in ATP to ADP ratios, affects reactions requiring these cofactors. Third, the redox status of the cell, measured by NADH to NAD+ ratios, influences dehydrogenase reactions. Finally, allosteric regulators can activate or inhibit specific enzymes, shifting the pathway direction. Let's look at how cellular conditions determine pathway direction. During fasting, high substrate availability and hormonal signals drive gluconeogenesis, pushing these reactions in the reverse glycolytic direction. Conversely, in the fed state, high glucose levels and insulin signaling favor glycolysis, driving these same reactions in the forward direction. To summarize the key points about steps 3 through 8 of gluconeogenesis, these steps use the same enzymes as glycolysis, but operate in reverse direction. They convert phosphoenolpyruvate to fructose 1,6-bisphosphate through a series of intermediates. The direction of these reactions is determined by cellular conditions, with energy and redox cofactors playing critical roles in establishing reaction direction. In step 9 of gluconeogenesis, fructose 1,6-bisphosphate is converted to fructose 6-phosphate through a hydrolysis reaction. This reaction is catalyzed by the enzyme fructose 1,6-bisphosphatase, also known as F16-bipase. Importantly, this step bypasses the irreversible phosphofructokinase step of glycolysis, creating a distinct pathway for gluconeogenesis. Let's examine this reaction in detail. Fructose 1,6-bisphosphate contains phosphate groups at positions 1 and 6. The enzyme fructose 1,6-bisphosphatase catalyzes the hydrolysis of the phosphate group at position 1. This reaction requires water as a reactant. The products are fructose 6-phosphate and inorganic phosphate. This enzymatic step is a key regulatory point in gluconeogenesis. Fructose 1,6-bisphosphatase is inhibited by AMP, which signals a low energy state in the cell. It is also inhibited by fructose 2,6-bisphosphate, which indicates that glycolysis should be active rather than gluconeogenesis. Let's examine the energetics of this reaction compared to its glycolytic counterpart. In gluconeogenesis, the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate requires only water. In contrast, the reverse reaction in glycolysis, catalyzed by phosphofructokinase, requires ATP. This difference in energy requirements makes the gluconeogenic direction more energetically favorable than the glycolytic reaction. To summarize, step 9 of gluconeogenesis converts fructose 1,6-bisphosphate to fructose 6-phosphate through hydrolysis. This reaction is catalyzed by fructose 1,6-bisphosphatase, which is inhibited by AMP and fructose 2,6-bisphosphate. Unlike the glycolytic counterpart, this step requires only water, not ATP, making it energetically favorable. This step is crucial as it bypasses the irreversible phosphofructokinase reaction of glycolysis, creating a distinct pathway for glucose synthesis. In this final step of gluconeogenesis, we'll examine how fructose 6-phosphate is converted to glucose, which can then be released into the bloodstream. The first part of this step involves the enzyme phosphoglucose isomerase, which catalyzes the reversible conversion of fructose 6-phosphate to glucose 6-phosphate. This isomerization reaction changes the structure from a ketose sugar to an aldose sugar without any energy requirement. It's the same enzyme that catalyzes this reaction in glycolysis, but in the reverse direction. The second and final unique step in gluconeogenesis is the hydrolysis of glucose 6-phosphate to produce free glucose. This reaction is catalyzed by glucose 6-phosphatase, which is located in the endoplasmic reticulum membrane. The enzyme hydrolyzes the phosphate bond, using water to release free glucose and inorganic phosphate. Critically, glucose 6-phosphatase has a very specific tissue distribution. It's present in the liver and kidney cells, but absent in most other tissues, including muscle. This tissue-specific distribution explains why only the liver and kidneys can release free glucose into the bloodstream. Although muscle cells can produce glucose 6-phosphate, they lack the enzyme needed to remove the phosphate group, so the glucose remains trapped inside the cell. The presence of glucose 6-phosphatase represents the final regulatory checkpoint in gluconeogenesis and has significant physiological importance. This enzyme is what distinguishes gluconeogenic tissues from non-gluconeogenic tissues. It's the final barrier that determines which organs can contribute glucose to the bloodstream. The importance of glucose 6 phosphatase is highlighted in conditions like glycogen storage disease type 1, also known as von Gierke disease, where a deficiency of this enzyme leads to severe hypoglycemia and enlarged liver. To summarize, the final step of gluconeogenesis involves two reactions. First, the conversion of fructose 6-phosphate to glucose 6-phosphate by phosphoglucose isomerase, and second, the hydrolysis of glucose 6-phosphate to free glucose by glucose 6-phosphatase. Energy requirements of gluconeogenesis. Gluconeogenesis requires significant energy input compared to glycolysis. While glycolysis produces 2 ATP molecules, gluconeogenesis consumes 6 ATP equivalents to produce 1 glucose molecule. Let's break down where this energy is consumed in the gluconeogenesis pathway. Step 1. The conversion of pyruvate to oxaloacetate by pyruvate carboxylase requires 1 ATP. Step 2. Converting oxaloacetate to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase uses 1 GTP, which is equivalent to 1 ATP. Steps 3 through 10. The remaining reversible glycolytic steps consume an additional 4 ATP equivalents. Adding these up, the total energy cost for synthesizing 1 glucose molecule from 2 pyruvate molecules is 6 ATP equivalents. From a thermodynamic perspective, this high energy cost makes perfect sense. Creating complex molecules requires energy input. The synthesis of glucose from simpler precursors is not energetically favorable without this energy investment. Breaking chemical bonds releases energy, while forming new bonds requires energy input. Gluconeogenesis involves forming many new bonds. This explains why glucose breakdown in glycolysis is spontaneous and releases energy, while glucose synthesis in gluconeogenesis requires energy input from the cell. In summary, gluconeogenesis requires 6 ATP equivalents to synthesize 1 glucose molecule, highlighting the significant energy investment required for this essential metabolic pathway. Insulin is a peptide hormone released by the pancreas in response to elevated blood glucose levels, typically after carbohydrate consumption. When insulin reaches the liver, it binds to insulin receptors on the cell surface, triggering a complex signaling cascade. Inside the cell, insulin activates a signaling cascade involving phosphoinositide 3 kinase, PI3K, and protein kinase B, AKT, which ultimately regulates transcription factors like FOXO1. Activated AKT phosphorylates FOXO1, preventing it from entering the nucleus. This inhibits the expression of key gluconeogenic enzymes like phosphoenol pyruvate carboxykinase and glucose 6-phosphatase, G6-PASE. While insulin inhibits gluconeogenesis, it simultaneously promotes glucose utilization through glycolysis and storage as glycogen. This coordinated regulation prevents futile cycling between gluconeogenesis and glycolysis, which would waste ATP. By inhibiting one pathway while activating the other, insulin ensures efficient energy utilization in the fed state. Cortisol and other glucocorticoids are critical hormones released during stress. They enhance gluconeogenesis by increasing the expression of key enzymes like phosphoenol pyruvate carboxykinase, glucose 6-phosphatase, and fructose 1,6-bisphosphatase. Epinephrine, also known as adrenaline, has similar effects to glucagon during fight-or-flight responses. It activates beta-adrenergic receptors, leading to increased cyclic AMP and protein kinase A activation. This ultimately promotes gluconeogenesis to rapidly increase blood glucose levels during stress or exercise. Growth hormone plays a key role in promoting gluconeogenesis during fasting and growth phases. It increases lipolysis, providing fatty acids as an alternative fuel source, which spares glucose and provides glycerol for gluconeogenesis. Additionally, it directly enhances the activity of gluconeogenic enzymes. Thyroid hormones, particularly T3, stimulate overall metabolic rate, including gluconeogenesis. They increase the transcription of key gluconeogenic enzymes and enhance mitochondrial function, which supports the energy requirements for gluconeogenesis. Multiple hormones work together to fine-tune glucose production based on various physiological needs. This integrated system ensures precise regulation of blood glucose across different physiological states like fasting, stress, exercise, and growth. Allosteric regulation is a key mechanism controlling gluconeogenesis based on the cell's energy status and metabolic needs. The cell's energy status directly influences gluconeogenic enzyme activity. High ATP levels signal energy abundance, activating gluconeogenesis. When ATP is high and AMP is low, key gluconeogenic enzymes are activated, promoting glucose production. Conversely, high AMP levels, indicating low energy status, inhibit gluconeogenesis to conserve resources. A critical allosteric regulator is acetyl-CoA, which activates the first committed enzyme of gluconeogenesis, pyruvate carboxylase. Acetyl-CoA is produced in abundance during fatty acid oxidation, particularly during fasting or low carbohydrate states. This creates an elegant feedback mechanism. When fats are being broken down for energy, acetyl-CoA accumulates and allosterically activates pyruvate carboxylase. This links fat metabolism directly to glucose production, ensuring the body can generate glucose during prolonged fasting. Another key allosteric regulator is fructose 2,6-bisphosphate, which controls the opposing activities of phosphofructokinase and fructose bisphosphatase. Fructose 2,6-bisphosphate inhibits fructose bisphosphatase, a key gluconeogenic enzyme that converts fructose 1,6-bisphosphate to fructose 6-phosphate. The levels of fructose 2,6-bisphosphate are controlled by hormones, primarily insulin and glucagon. Insulin increases fructose 2,6-bisphosphate levels, inhibiting gluconeogenesis. Conversely, glucagon decreases fructose 2,6-bisphosphate, activating gluconeogenesis. These allosteric regulatory mechanisms ensure that gluconeogenesis responds appropriately to the body's energy status, available fuel sources, and hormonal signals. During the first 24 hours of fasting, the body primarily relies on liver glycogen stores to maintain blood glucose levels. After glycogen is depleted, around 24 hours into fasting, gluconeogenesis becomes the primary source of glucose. During this phase, protein breakdown provides amino acids that serve as substrates for gluconeogenesis in the liver. After 2 to 3 days of fasting, the body shifts to ketosis. Fatty acids from adipose tissue are converted to ketone bodies in the liver. The brain adapts to use these ketones for energy, reducing glucose demand by about 75%. This protein-sparing effect reduces the need for gluconeogenesis from amino acids, preserving vital muscle tissue. These metabolic adaptations allow humans to survive extended periods of fasting, sometimes lasting several weeks. The shift to ketone metabolism and the resulting decrease in gluconeogenesis preserves vital protein structures while efficiently utilizing fat stores for energy. To summarize, fasting fuel usage progresses from glycogen to protein to fat. Gluconeogenesis is critical during early fasting, but decreases as ketosis develops. This protein-sparing effect of ketone bodies reduces gluconeogenesis requirements and is essential for long-term survival during extended fasting. In diabetes, gluconeogenesis becomes dysregulated. While normal metabolism maintains a balanced rate of glucose production, diabetic patients experience excessive hepatic glucose output. The root cause of excessive gluconeogenesis in diabetes is a hormonal imbalance. In healthy individuals, insulin and glucagon maintain a balanced ratio. In diabetes, insulin action is diminished while glucagon signaling is enhanced. This hormonal imbalance leads to upregulation of key gluconeogenic enzymes. Reduced insulin signaling removes the inhibitory effects on enzymes like pyruvate carboxylase PFP, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. Meanwhile, increased glucagon promotes their activity. A key feature of diabetes is hyperglycemia despite fasting. In healthy individuals, blood glucose stays within a narrow range. In diabetes, excessive gluconeogenesis leads to elevated blood glucose even after hours without food. This explains why many diabetic patients wake up with high blood glucose levels despite not eating overnight. Metformin, one of the most prescribed medications for type 2 diabetes, works primarily by reducing hepatic gluconeogenesis. It acts through several mechanisms: activating the AMPK pathway, decreasing cAMP signaling, reducing cellular energy through inhibition of complex 1 in mitochondria and suppressing the expression of gluconeogenic genes. Diabetes features a vicious cycle where hyperglycemia worsens insulin resistance, which reduces glucose uptake by peripheral tissues, further increasing gluconeogenesis and resulting in more hyperglycemia. This self-reinforcing cycle explains why untreated diabetes typically worsens over time, with progressively increasing insulin resistance and gluconeogenesis. Genetic disorders of gluconeogenesis reveal the critical importance of this pathway for normal metabolism. Von Gierke disease, also known as glycogen storage disease type 1, is caused by a deficiency in glucose 6-phosphatase. This enzyme catalyzes the final step in both gluconeogenesis and glycogenolysis, converting glucose 6-phosphate to free glucose. Without glucose 6-phosphatase, the body cannot release glucose from the liver into the bloodstream, even when glycogen is broken down. This leads to several serious clinical consequences. Patients experience severe hypoglycemia during fasting periods as they cannot mobilize glucose from glycogen or create it through gluconeogenesis. Glycogen and glucose 6-phosphate accumulate in the liver, causing hepatomegaly, an enlarged liver that can be several times its normal size. Von Gierke disease is the most common, but other enzyme deficiencies in the gluconeogenesis pathway also cause significant metabolic disorders. Pyruvate carboxylase deficiency affects the first committed step of gluconeogenesis, preventing the conversion of pyruvate to oxaloacetate. Other rare disorders include phosphoenol pyruvate carboxykinase deficiency and fructose 1,6-bisphosphatase deficiency, each blocking a specific step in the pathway. These genetic disorders highlight the essential nature of gluconeogenesis for normal metabolism and glucose homeostasis. Each enzyme deficiency creates a unique metabolic signature, but all demonstrate how critical maintaining glucose levels is for human health. Understanding these disorders provides valuable insights into the biochemical pathways that maintain glucose homeostasis. During intense exercise, muscles rely heavily on glycolysis to generate ATP rapidly. This anaerobic process produces lactate, especially when oxygen becomes limited. Lactate is then released into the bloodstream and transported to the liver. In the liver, lactate is converted back to glucose through gluconeogenesis. This newly synthesized glucose can then return to the muscles, completing what's known as the Cori cycle. This metabolic cooperation between muscle and liver is crucial for clearing lactate and maintaining glucose homeostasis during recovery. Endurance training increases the liver's capacity for gluconeogenesis. Trained athletes have higher concentrations of key gluconeogenic enzymes like pepsi-K and G6-PACE. This enhanced gluconeogenic capacity provides several advantages for endurance athletes. These include faster lactate clearance, improved glucose availability during prolonged exercise, and enhanced recovery between training sessions. After exercise, gluconeogenesis continues to play a vital role in recovery. The process helps replenish glycogen stores in both the liver and muscles. Proper nutrition can significantly enhance this recovery process. Consuming carbohydrates post-exercise accelerates glycogen synthesis, while protein intake provides amino acids for gluconeogenesis. The optimal nutrition window is within 30 to 60 minutes after exercise. In summary, gluconeogenesis bridges exercise and recovery by clearing lactate, restoring glucose, and supporting glycogen replenishment.